guys, you can hear me now?
Yeah, I hear you clearly.
Yes.
Thank you.
Right, great. All right, thank you to the MC, our keynote speaker, Dr. Tjeerd Dijkstra, fellow conference participant, ladies and gentlemen. Good morning or good afternoon, depending on where you are, and Assalamualaikum warahmatullahi wabarakatuh. So first of all, let me just introduce myself. I am Norhidayah Hamzah from School of Computer Science and Engineering, Taylor's University, Malaysia. We have a lot of computing and engineering-based programs, particularly like artificial intelligence, data science, robotic programming, data visualizations, and such. And we are always open for collaboration in any way possible. It is my honor to be the moderator of this session on synergistic combinations from high throughput drug screening. We will have 50 minutes of keynote speech followed by 20 minutes Q&A session towards the end of this talk. So if you have any questions to ask, please feel free to directly ask our speaker using the raise hand feature, or you can post your questions in the chat room. We will attend to the questions after the keynote speech ended. Without further ado, let me just introduce you to our keynote speaker for this session. Dr. Tjeerd Dijkstra is a researcher at Translational Bioinformatics, University of Tubingen, and Department of Women's Health of the University Clinic Tubingen. Before serving in his current employment, he was a research scientist in renowned universities in the Netherlands and USA. He is the author and co-author of more than 50 publications and patents, of which majority of it are peer-reviewed publications. Dr. Dijkstra has also conducted extensive research in areas such as gene expression, posture, and motion perception, which led him to secure numerous national and international fundings, one of which was awarded by the National Institute of Health USA with a grant amount of $1.95 million in 2014. He is currently a co-principal investigator in a research grant amounting to more than 500,000 euros. He has been teaching bioinformatics modules since 1992 and has supervised nearly 30 postgraduate students in master's and PhD in the field of physics, computer science, and bioinformatics. Ladies and gentlemen, please welcome Dr. Tjeerd Dijkstra to deliver his keynote speech entitled Synergistic Combinations from High Throughput Drug Screening. The session is yours, Dr. Dijkstra.
Well, thank you very much. My head is glowing after so much praise, I must say. Also would like to thank Rido Rahmadi for inviting me to give this keynote talk. And selamat siang, selamat orang Indonesia dan Malaysia. Kalau bicara pelan-pelan, juga bisa tanya pertanyaan dalam bahasa Indonesia, tapi mesti bicara start really pedestrian, reviewing many of the kind of classical developments in the field. That's the first part. The second part is you could say current developments, things we're working on right now and that haven't been published yet. So with that said, let me try to get to the talk. Share. Yes, and then I need to. How do I play this now? Okay. Everybody can see that?
Yep, we can see the slide.
Yeah, okay. Technology sometimes. I've had troubles with Zoom before, so that's why sometimes strange things happen. Okay, and one other detail I want to point out, teeny tiny detail, is in the bottom here, all slides are numbered. So if you have a question about a specific slide, then please write that number down and we'll get to it in the end. Okay. Why doesn't it work? Okay. So as I said, there are 3 parts to my talk. This is the first part, a kind of historical overview. And the motivation for all of this is, change a little bit on the layout of the screen. Sorry, it's beginners problems. Do I get back? This wasn't what I wanted, so let me go back. Okay. Sorry about this. Zoom beginner.
It's okay. We
For some
still
reason,
have
I
time.
cannot go back.
No
Yeah,
worries.
for some reason, I cannot go back once I share my screen, which is somewhat annoying. Okay, I'm still screen sharing. Okay, so that's good. All right, so for some reason, you cannot go back while you're in Zoom. Must be a feature. Okay, anyway, the motivation of this is what's called high throughput interaction screening. And I'm introducing the technology here. What you have is typically a plate, as it's called. This is 96 well format. In the research I'll talk about, there are 384 wells, and there's even well formats with over 1,500 of them. And what you do is you dispense a library, typically some cell types. In our research, those are human cell lines, but it could also be a bacterium or a yeast or something. Then you dispense your reagent, your compound. In our case, those are anti-cancer drugs, but it could be any other compound that you want to screen the activity of. And then in the third step here, the readout, you have some kind of detector, typically a laser, where you read out an optical signal. Later, I'll talk about another type of readout, which is a kind of gene expression. Now, this is quite commonly used. One example is drug compounds, where compounds are screened against cell types of interest. So talk about the recent history, right? It's known that that the coronavirus enters cells through the ACE receptors. So that I'm sure there has been a lot of screening going on to check the activity of compounds relative to cells that have this ACE surface receptor, right? We work in this field, and we also work somewhat in this kind of inverse field. So here it's good when you get an effect, typically. In environmental health, it's typically bad when you get an effect. So what you do, you screen some standard organisms that are common in soil or in water, and then you hope that compounds have no activity against them, meaning that they're not poisonous. Okay, so, and now the question you ask is, oh yeah, one other review is drug receptor theory. If you've had chemistry or pharmacology, this is probably very basic, but I'll introduce it here to get everybody on the same page. Imagine that you have some kind of receptor. In this case, it's as one particular example, it's a cell membrane bound G protein coupled receptor. You have neurotransmitter, that's the drug here, and this receptor is still free. That's here, and then when they bind together, you get a complex. Everybody's in the waiting room pops up on my screen, so that's a bit of a distraction. I'm sorry. Okay, so right, so the free drug I denote as XA. The total number of receptors n tot, and the number in the complex is Na. So the free is then n tot minus Na. And then you can write reactions, that times the reaction constant is the forward reaction going that way, and that concentration times that reaction rate is the reverse reaction. Now in chemical equilibrium, the two have to be equal. So you write this, and then it's a trivial algebraic manipulation to solve that for the fraction of occupied receptor, right? Which depends on XA in this kind of way. This goes by many different names in the field. Now, rewriting this slightly in the notation that we'll use from now on, I write Y is the response, X, I keep that as kind of the dose, the amount of compound that you add, and E is the equilibrium constant, which is written here. So it's not to be confounded with the number E, 2.7 natural number. So in pictures, this is how it looks. Right, you get this sigmoid shaped thing. And I generalize that a bit hand waving, where instead of this fixed slope, you could say -1. I now give it's a parameter, which is here the slope of this thing. This E is known as the EC50 concentration, which is set at 0 by the scaling here. And there is a lower and upper threshold, although those might be fixed depending on your exact assay. Okay. That's what is now the problem. Given the dose response curves for 2 agents, what response can one expect to a mixture of agents under the assumption of no interaction? Right. So, put another way, what is a good null model of no interaction? And if you have such a good model, and we'll talk a lot about that in the next half hour, then synergy is when you have more effect than predicted from this interaction model. Antagonism would be less, and what's known as additivity is basically the same as what you expect. Now, this goes by different names in the different fields. In pharmacology, it's combination therapy. In toxicology, it's known as mixture toxicology, right? The combination of drugs. You have a mixture of drugs. And in statistics, especially industrial statistics, this is sometimes known as response surface methodology. Okay, now some bad news, I guess, or this field has a very confusing history, and I'll slightly talk about that further down. Here I just cite two influential reviews, right? One from the late 80s that unfortunately there's considerable confusion on this question of synergy, so much that for the past 30 years or more, it has almost been an established custom to begin reviews on synergy with a discussion on the prevailing confused and archaic state of affairs. And this is from Chu. It's hard to find any other field of biomedical science that has more controversy and confusion than drug combinations. So much for history. Okay, now we go back a lot in time. Among the first publications on this field, that's by a German professor named Loewe. The original publication is from the 20s in German. It was more or less republished in the 50s in English. And I kind of like to show these classical pictures because they give a lot of intuitive insight, and then I'll modernize them for our own discussion. So here you have the dose axis of compound A, and in blue is kind of my modern notation. So I'll call it X1 later on, but it's called A here. The other one's called B, X2, and then here's the effect. Call it Y. And then imagine you have this response surface, right? So for only dose X1, you will get this S-shaped curve. For only X2, you get that one here. And then for any mixture, you get anything on this kind of dashed surface in between, right? Which I will call H, is a function H of X1 and X2. And if the surface looks like this, then if you slice it at a certain level, in this case the 50% effect level, then you'll get a straight line cross section. And later on, we'll call cross sections isoballs. I'll explain a little bit of that. That's the kind of way things are called in this field. So this is one case, kind of very aptly described as tenth sale in the classical publication. There are other cases when you get synergy and antagonism. So synergy is when you get more effect than you could have expected based on that previous hypothesis, right? So then what you get is when you do the same thing, to make the cross-section here at level 50, then you get this thing that bows inward, closer to the origin, right? So cross-section bows inward, and then there is synergy because you have more effect than you could have expected from each of the compound alone. And antagonism is the opposite, which is when it's further away. So now if you only focus on this 50% level and put all three curves in, what you get is this view, right? This is the AB plane at level 50, right? Additivity is just a straight line diagonal, synergy bows inward, and antagonism you bow outward. Okay, that's that. Now, moving on to the recent era, this is my visualization of it. I have agents 1 and 2, colored red and blue here. Same here, red and blue. These are the kind of monotherapeutic responses, you could say, right? Response versus dose. Same here. And then when you plot it, here it looks like here. Now, first off, from now on, we'll exclusively have responses that go down with increasing dose. Almost always, right, you want to achieve an effect of cell death or growth inhibition, which you more conventionally plot, right, with increasing dose going downward. So we'll do that from now on exclusively. And then here are the so-called isoballs plotted, right? From now on, I'll call these iso response curve isoballs. And here at the 10% effect level, 20%, 30, 40, 50, etc., all the way up to 90, right? And that's then generated under assumption of no interaction, right? What I've done is I've taken this, and I'll talk about what I exactly did, and then generate this response service. I generated that. So let's switch to some mathematics here. Given the dose response models for each of the agents individually, right? Response is a function H1, the Hill model of dose X1, same for 2. Then the goal is to construct a null model, which I indicate with 0, which is a function of X1 and X2, with the obvious constraint, right, that on the boundary, right, if this one's 0, then I should get that one back. And if that one's 0, I should get that one back. That's written here. Okay. Now, first, a kind of classical derivation that I try to put into mathematics as good as possible. The original formulation is purely text, and it's also a wrong formulation, but it's been very influential in the field. It is from the late 80s by a person in Birnbaum. So Loewe proposed this principle that an agent does not interact with itself. That seems rather obvious, right? You have one compound, you divide it in 2 parts. There's no interaction between those 2 parts, right? Okay. Thus, when you have a certain dose X1 from agent 1, plus you take the equivalent from agent 2, but translate it into a dose of X1 that has the same effect, then that should also be non-interactive, right? Because you just translate it from one compound to the other in terms of the effect. Okay, mathematics, we take a fixed reference level star, that was the 50% level that we saw in the previous pictures by Loewe. Then we have y star is this Hill function of x1 star, and same for x2. Now take a dose that's smaller than this x1 star. How much of x2 do we need? How much of x2 do we need to achieve the effect y star? The dose ratio allows one to translate, right? So this ratio of stars then allows one to translate and kind of make up the effect, right? And that then leads to the same y star. And now we can invert this, right? And then divide by x1 star, and we get this kind of constraint equation, which looks like a the line that we just that I just showed, right? And it's called the generalizable equation. The frustrating part is that I cannot go back. Oh shit, this was a bad idea. Okay, I can go back, but right, this line basically. Right? That's the one. So this one, this would be x2 star, and this would be x1 star here, right? Okay. Okay. And then, okay. All right. Now there is a problem with Loewe additivity, and that was known, but we highlighted here. So what we've done with this kind of translation is kind of laid out here. You take a dose, so both doses are plotted on the same axis, x1 and x2, and these curves are the response curves, the hill curves. So I take x1, and that has that effect. And then I look at the dose response curve h2, and I can translate that in a kind of effect equivalent amount of x2. Right. So in mathematics, this is how it looks. I take x1, I take that red curve h1, I then take the inverse of h2, but these curves are always monotonic, so I can do that. This gives me an equivalent dose. And I can do the same one for x1, right? Starting with x2, took the z effect, that, and then I get that one there. Okay. So I can do that. Then what I can do, I can construct two null models. I can construct one from agent 1 plus the equivalent dose of agent 2. And remember, this is all x1, so it doesn't interact with itself. And now I only have x1, so I know how to get an effect from that. That's just h1. That's one possible null model. I can, of course, do the opposite, right? Starting from x2 and translating x1 into its x2 equivalent, I get this one. And then substituting the expressions we had before, we get this impressive mathematics, right? But the point is now that the labels 1 and 2 are obviously arbitrary, so they need to be equal. Right? So equality of the two null reference models is this equation, which we called the Loewe additivity consistency constraint. This has to be true for any x1 and x2. And what we showed in a recent paper is for this to hold, what you have to have is that x1 and x2 need to be proportional to each other. Right, so there's a constant c, arbitrary constant, a positive constant, then these two are equal. And that means when you can scale the two agents, that means in log units, you just shift them. Right, so this is actually in log units, right? So these two are not parallel. Actually, I think they are parallel, if I recall correctly. So then it works. If not, then it doesn't work. And we show some examples of that later on. Right? So this was kind of known since the mid-90s, but we are the first one to provide a mathematical proof of that thing. Okay. So now we have two, I present a new null reference model, the generalizable model we've seen. So now what I do is instead of this kind of fixed level x1 star and x2 star, I make it any level depending on y, right? So I need the inverse hill of that. That is an invertible function. It's just that when you do this for two different hill functions, you cannot explicitly solve it. So you need a numerical root finder for the solution, but I mean, you can do that, it's just work. What we propose is an alternative model, which is basically a very simple linear mixture of these two. And there are some reasons why we did that, but I won't go over it now. That would be too much. All right, so kind of brute force approach in a certain sense. But for one thing, it's explicit. You don't need a numerical root finder to solve this one. Okay. So what are the predictions for these two different models? Here are some cases, one where the slopes are unequal, right? So this one's obviously more steep than that one. One where the asymptotic levels are different. This one goes to 0, and this one has an asymptote somewhere, I think 0.3. And this is a case where they're both different. So I'm just exploring the theoretical differences. No data yet, right? Slopes are different, the asymptotic levels are different, and here both are different. And then what you have is you always get these straight line isoballs for the GI model. Right, so this is dose of x1, dose of x2, and here are the various isoballs, 0.1 all the way to 0.9. Here they're the same here, and they kind of fan out at a certain point. There are some details I gloss over here, but a general point. Now for the explicit mean model, those isoballs are not always straight, right? But in general, they're closer to the origin, right? The 0.3 is shifted from there to here, for instance, and the 0.2 is shifted from here to there, for instance, right? So they're always closer to the origin, meaning that it's lying actually right above the other one, if I recall that correctly. I mean, it's more conservative, and put it that way. And we'll see that later on also. Okay, that was that. So I took you from the 20s of the last century all the way to last year in a really bird's eye view of the field of synergy. Okay, now let's benchmark these data using some real data. Now we used two data sets for that. A problem in this field, and it's probably one of the reasons why this confusion has persisted so long, it's not obvious what a ground truth is. It's difficult to generate data sets where you really know that there is synergy or antagonism, right? To the best of my knowledge, there are no such data sets available. We talk about later our attempt to mitigate that and how far we are with that. But as of when we started this work in 2016, there simply were no data sets. So what we did, we took two data sets where the classification was made subjectively by someone, but it was not made by us. Right? So we took the labels and the kind of raw data from other sources with the big right question mark that the ground truth was a label that someone else provided. Okay. Having said that, and there's one which we call the Matthews-Greiner or Yadav data set. Matthews-Greiner originally collected the data. Yadav provided the label. It is anti-cancer drug combination, this particular cell line. The phenotype is cell viability. We'll talk a little bit about this particular assay later on. And right classification was done by visual inspection, provided by Yadav. And the other data set is by Cockle from 2011 publication. And here the phenotype is here really cell death, and here it's growth inhibition. So basically you shine a laser through those things, and then DNA absorbs this wavelength, and then the less light you get through, the more DNA there is, so the more cells there are. That's the kind of simple mapping. The other things you can ignore for this setting. So now, as I said, we compare two models, the kind of standard general isoball model and our new model, which you call the mean model, or sometimes the explicit mean model. We're somewhat inconsistent sometimes in labeling the figures. I'm sorry about that. How do we evaluate model performance? Well, we have the measured effect, right? This kind of visualization of the raw data. Then we take each of the conditional ones, right? Fit a Hill model to that, and then generate the surface with the two models that I just showed, right? So this is kind of a model surface. And then we take the difference between the two, called deviance here, and then we just kind of average that. We don't look for kind of complicated effects where you could have synergy for one combination and antagonism or additivity for others. That's complication that we ignore for now. So we take this difference, and then initially we only investigate drug combinations labeled additive. So additive means that these two should be very close. So if you have a good null reference model, then this model should be close to the measured effect, and the deviance should be 0, ideally, right? So the mean squared error should also be 0 then. Smaller error is better. So how does that then work in practice? Here are the two data sets, Matthew Greiner and Cockle. Here is our model plotted versus the same on the GI model. Now first, this data set, almost all data points are above the diagonal, right? Here's an exception. And so our error is clearly smaller than that one from Cockle. Here the differences are a bit more subtle, because there are these kind of data points that really draw attention, where we actually do worse. But if you look carefully, the vast majority of data points are here, right? Here you see the histograms, right? And they're actually above the diagonal. So in both cases, our model does a lot better, and some kind of statistical test, Wilkinson signed red test, right, is highly significant. We do better on both. Okay, that was on the things labeled as additive. Now we also have labeled synergistic and antagonistic, and I'll briefly show that. This is a very complex plot, and it has more information than what we need. Right, this is kind of the raw data, and with some fantasy, you can see some bowing in here, but probably a lot of fantasy. Here you have the kind of raw data from, right, if this compound is 0, so only tar, come to dose, and then we fit that Hill model to it. This is the reverse, but plotted upside down. Honestly forgot the reason why we did that, but there was some smartness to it. And with that, we generate a null references that's not shown. And then the difference is shown here for all the models that we tested. That's many more than I'm discussing now. Here's the generalizable one, and here's the explicit mean one. Red means synergistic at that location. Blue means right, antagonistic at that location. And you can see that, right, the generalizable predicts a higher synergy here than we do actually, but both are above threshold. Okay, other case, antagonistic. Here again, Fen DIC combination. Well, you need probably a lot of fantasy to see that these bow the other way, bow outwards, but okay. Here's again dose response curve and the fit. And here are the two models. Both show clearly antagonism, right? Our case, our model a bit more than the generalizable one. But these are just two cases. We have hundreds of those. So how does that work in overall? Using Kendall's rank correlation coefficient, we see that, and again, I didn't get rid of the other models in these plots, is so compare this row here with this row here. We do a bit better here, and we clearly do better here relative to that one, right? We're not always the best. There's one other model that in this particular case is better. So, right, we beat the generalizable model in both cases. Okay. Now, what is exciting for me to talk about, I think this is the first time I talk about it, our new developments. And I think, how am I doing time-wise? I still have 20 minutes, right?
Yeah, more or less
okay,
20
okay,
minutes.
good. Okay, okay. Then I was worried that I wouldn't make it to the end and not have time to talk about that. Okay, good. So this new developments. Two things. As I noticed, there are no good benchmark data, so we try to fill that gap with a kind of experiment that's specifically tailored to be a benchmark data set for synergy prediction. So far, all the data sets have been collected for another purpose, screening anti-cancer drugs, of screening antifungal drugs for the two data sets that I discussed, but for other data sets, that's true also. And the second is that we use a very modern machine learning technique called Gaussian Processes for synergy prediction. Okay, to dive right in. The first thing is to give the background of that is to predict synergy, right? So far, what I've talked about is given measurements, can we have to, how do you establish that a combination is synergistic, right? But of course, ideally, you would not want to do the experiment to begin with, or at least screen for interesting candidates, compounds that might be synergistic, and only screen those, right? As opposed to kind of going blind. And that there is more and more activity in this field. It's just one paper on a slightly different topic, but the same idea, right? Can one predict synergy just given the molecular properties? And here's a deep learning approach to discovering a single antibiotic, right? No combinations. But so they encoded the structural properties of small molecules and used those to virtually screen for new antibiotics, and came up with a compound that they then actually verified that worked as an antibiotic. So quite an achievement to do that in silico, as opposed to screening millions of compounds. Okay, now before one gets too enthusiastic, there is one complication, and this is not the best slide I could find, but I was somewhat pressed for time. It's at least one example. So whether combinations are synergistic or not sometimes depends on the cell type itself. So combinations that are synergistic in one cell type have sometimes the opposite effect on other cell types, right? Making it a very complex interaction matrix, right? So it's not purely a property of the molecule, it's also a property of your target. So here's an example of that from a recent paper. So beta observe is some quantity that captures synergy, positive meaning synergistic, and below zero, where there's some compounds here, and here means antagonistic, although they're not too antagonistic relative to synergistic, but whatever. And the combinations have a letter A, and the combination A is dabrafenib and gobimitinib. Man, these things are hard to pronounce always. Okay, now I've highlighted a few examples. For instance, combination P is weakly antagonistic in these two cell lines, SKMEL5 and WM9A3B, whereas it's weakly synergistic in these two, A375 and SKMEL28, right? So it depends. That's not always true, right? I think if you look carefully, K is always among the good ones if it's there, right? K is always very synergistic, except for this one where it probably didn't show up. But I mean, it shows you that synergy is a complicated concept, right? And it can, the actual how synergistic something is can depend on the actual cell type. And this is another example of synergy prediction, a very recent paper from the Barabasi group in Nature Communications. And here they used kind of drug databases. So this is, you could say, even higher level than my approach. Right, my approach is for screening, so it uses cell types. Of course, if you make an actual drug combination that you apply in humans, there comes a whole different level of complexity, right, through it, like pharmacodynamics and how drugs are metabolized and taken up by the body, and etc., etc. But here they used combinations that were known from the literature and adverse drug interactions that were known from the literature, right, from just drug databases. And what they did is they looked at network properties. So you construct a protein interaction network. Those are those nodes and those interactions. Those are often known from screening, and you can download those from databases. And then you know that a certain disease afflicts a certain part of this protein interaction network. It's highlighted as red and a kind of pinkish color here. Right, and then they looked which part of the network is attacked by drug A and drug B. This assumes you already have a lot of knowledge, right? But they curated their databases to have that. Now, if you have this case where the activity of the two drugs overlap, of course they have to overlap with the disease network, otherwise they probably don't do anything, right? Then you don't get an awful lot of effect, right? You find very few of the good combinations and a lot of the bad combinations. However, when the two drugs kind of are independent and attack different parts of the disease network, so you could say complementary, then you have a lot of the good ones and a lot less of the bad ones, right, relative to random. So it is a very different approach to predicting synergy, but might be successful too. But there are no data at the cell level, at the cell line level. So how do we do that? Okay. So this is what we did. It's an experiment in collaboration with Nick Lyons from the Broad Institute in Boston. And we are collecting a benchmark data set with both phenotype, which is cell death here, cell viability, and gene expression, a so-called L10001 assay, and I'll explain in a minute what that is. We're only at step 1. It's a 2-step procedure, and given and that these things are relatively costly, we need to optimize our experiment really in order not to waste any resources. So what we did so far, we've screened 20 compounds with 12 doses each, so a very wide range with 3 replicates. And we used 3 cell lines in the first iteration because we know that cell lines can react differently, right? And we've used that to optimize. We've actually redone this step. There were some initial problems, and we're still analyzing, you could say, step 1A we've done, step 1B that we're at. And the plan is then to select a subset of these compounds that work really very well in both assays and in all the cell types, and then run a full combination assay for a subset of those combinations. So how does this technically work? Right, in the first we use the cell viability assay, and what it is, it's based on you could say the energy that's in a cell that's provided by ATP, adenosine 3-phosphate. And in live cells, there's a lot of it available, and in dead cells, it's rapidly degraded. And what you do is you have a reaction that emits light, but it uses ATP, right? And this chemical reaction will emit light, and the light that you detect. That works well in theory, but it has trouble with compounds that act on the ATP mechanism or that change cell size, because bigger cells have more ATP. And then in the second round, we've done it slightly differently, which seems to give better results, but this is one way to do it. This is what we kept. We have a gene expression array, which is called L1K, L1000 or L1K, and it's based on a miniaturized microarray. That's how you can view it. The assay is cheaper than microarrays and a lot cheaper than RNA-seq, but the downside is it can only quantify around 1,000 landmark genes, 974, whereas the human genome has, around 20,000 genes. So that's a trade-off that you make. However, we believe that those landmark genes were chosen very carefully, so that each pathway, you could say, is covered by several of these genes. So in general, I would say you capture 80 or 90% of the variability of response of a cell to stimulation. And this is the technical aspects. I won't go in it. It's a very fancy technique, and it's very cost effective. That's probably because, right, running all these those combinations runs you into many different assays. Okay, then the last part. Okay, yeah, 10 minutes. Gaussian processes for synergy. So this is very different. The first part is experimental, collecting some benchmark data. The second part is new theoretical developments using a non-parametric regression. So I showed it, but I'm highlighting it once more, that our approach so far, and in honesty, almost everyone else in the field has been based on hill models, right? So both figures that are up here I've shown before. I just highlight a different aspect, right? This null reference. So what you do here is you fit a hill model and you fit a hill model there. And here it's explicit, here's the hill model, and there's another hill model fit, right? And you use that to then generate a reference surface. But can we do better with a non-parametric approach, right? These things are parametric, they're not very flexible. And another thing is, which I've ignored so far, can we include time beside compound dose as another stimulus dimension in the same framework, right? I always plot the things as a function of dose, but what you effectively do is you have these wells, you add the compounds, and then typically you wait, if it's a destructive readout, you wait 24 hours or 48 hours, or sometimes even longer, and then you measure your effect. Right? There are now more and more techniques where you can kind of almost continuously measure, and we actually have that for the Cockle data set, where they did yeast and they measured optical density. Right? You can shine the laser. I think they did it every 45 minutes, but collecting a lot of data, and there's more of that. The L1000 readout or gene expression readouts will always be destructive, but there are many phenotypical readouts which now have time also as a component. Right? And of course, you can construct these null references for each time instant, but it would be elegant to have an integrative approach. Okay. With that said, let's dive into Gaussian processes. I'm slightly reviewing it here, but this is an extremely high speed review, so I hope you have some familiarity with it. Right, it's a non-parametric regression with the covariance structure determined by the kernel. So the kernel is where the action is. And the most common kernel is the so-called linear squared exponential kernel, which looks like a Gaussian, but it's a smoothing, right? What you exploit here is the property that this is an extremely smooth function, infinitely differentiable in x and x prime, right? And x and x prime are two doses. And there are two hyperparameters here. Sigma is the scale, kind of scales the response. And then L is the length scale of the dose that scales the dose, basically how wiggly these lines look. Right, and you can view this as a prior on the covariance functions, and then you can do random draws. And if you've read any article about GPs, you've probably seen pictures like this, right? This is a 3 different random draws from a prior that has that kernel, right? And you get these smooth wavy functions. Right, this is dose, this is response. This is how it look. Okay, now what did we do? This standard kernel works good in general, but our specific setting, we found the logarithmic kernel to work better, right? So almost always in chemistry and pharmacology, the effect of a compound is in terms of the log, right? You're probably all familiar with the pH scale of acidity, right? That's a concentration of free hydrogen, positive hydrogen atoms in, let's say, water. And that's also a logarithmic scale because that's how the activity scales, right? Typically with the log. So in order to kind of put that into our model, we designed this kind of new logarithmic kernel. And it's basically, right, the difference in the logs. Of course, you cannot take directly the log of X because concentration 0, dose 0, is a very important point, right? And then this thing would explode. So what we do is we take log 1 plus x divided by L being again the length scale. It's not obvious, and maybe directly that it's a length scale. What you can do is if you take x and x prime to be very small, then you know from elementary algebra that you can approximate this by x divided by L. And then in that limit, right, with x and x prime being very small, relative to L. Then this logarithmic kernel kind of reduces to the linear one, and then you see that the interpretation as a length scale is correct, right? And maybe as a side note, for those who are really paying attention, right? But the Hill model was kind of written directly in terms of X, right? Well, actually in practice, if you fit it, what most fitting routines do, they write it this way, right? So the slope then becomes just an, you could say, a regression parameter in terms of log X, right? It's basically logistic regression in terms of log X with an offset, right? So the Hill model can also be viewed as a log logistic model. Okay. So how does this look with this new fancy kernel? I've plotted here 4 sets of 3 samples. This is basically the figure that you saw before, exact same one, and they're identical realizations, right? So the same. I reinitialized the random, so they're identical. I fudged the random number generator, put it that way. So this is with our linear kernel. The bottom ones are with this fancy new logarithmic kernel, and here I've varied the length scale to give an idea, right? This looks like wavy lines. If you decrease the length scale, it becomes more wiggly. And then the logarithmic kernel, plotted in function of a linear dose, what you only get some wiggles in the beginning, and later it becomes rather flat. Of course, you increase the length scale, it becomes a little bit more wiggly in the beginning. Of course, this is plotted in linear dose, right? If you plot it in logarithmic dose, then this is how it looks, right? So this is plotted as a function of log 1 plus x divided by L, right? This is the argument of the kernel. Then, of course, I mean, these are kind of homogeneous, right? And then the linear ones have all the action at the right-hand side due to the compression of the log, kind of reverse pattern as before. Okay. So now how well does it do in practice?
I'm sorry to interrupt, Dr. Dijkstra. We have another 5 minutes.
Okay, I think I had 44 slides or 43. So
yep, I think that can
work,
cover
right?
it. Yep.
Okay, well, I'm proud of myself. Almost there, yeah. So this is some data that are actually simulated from a Hill model. So kind of stacking the odds against the GP model. They're not real data, but they're a well-known benchmark data set, kind of which many people use from this Greco paper. So the data are simulated from a Hill model. So of course the Hill model should do very well, but as you can see, the Gaussian process model also does very well, right? Maybe it's a bit too good, but definitely not worse, right? And we need to benchmark this more with real data, but we simply haven't had time. Okay. Now, this is the same kind of simulated data from that Greco thing. Again, here are the null model contours, right? This is dose compound 1, dose compound 2. This is a smoothed version of the real data. We can use our GP now to smooth the data, right? Here, and then this is this difference again. Now, this was generated under synergy, and positives mean synergy here. Even yellow is weakly synergy, light green is a bit more synergy. So on average, it's indeed synergistic as you would have, right? Now, this was not generated under a Loewe model, but under we also took the opportunity to use a very recent model, which is called the hand model. And that's shown here very briefly. So these equations I showed before, right? This is this underlying this Loewe model where you take all of dose of compound 2, translate it through this mechanism to equivalent dose of 1, add it to the one that you already have and say, okay, then H1 will give me the effect, right? And this is the kind of way, and then we had this ambiguity depending on where you started. Now, this is a solution that Hand published already 20 years ago, but it has been largely forgotten and was recently revived in a paper by Sinsger, and we use that. Is what you do is you do it kind of locally or differentially. What you do is you take, let's say, it's arbitrary, but you start with A. So I'm sorry for the slide change of notation here. I took it from different things. Here it's called A, that's 1, and here it's called B, that's our X2. You start with A1, translate that, then you take a little bit of B and use that, then you use A again, B again, A, B. So each time you translate a little bit from one to the other and add the effect to it, right? That's a very hand-wavy description of the hand model. Okay, and once you do that, and you can also integrate time. Here are some real data. This is from, I think, a TER-CIS experiment. Now, kind of ignored, but directly when you add the drug, there's not going to be a heck of a lot of effect, right? It's effectively flat. And of course, your GP model just then effectively makes a straight line, right? Now, after 15 hours here, you get a little bit of effect. And this is after 24 hours, right? You get a lot of effect, depending on the dose, right? This is for one compound. This is the other compound. Probably a bit of bigger effect, right? And the GP is smoothing over it. And this is the synergy at the beginning and at 24 hours, right? I'm ignoring all the steps in between that we generate a reference surface, we smooth the whole surface, subtract, that's what you see here. So initially there's not much effect. There's a delay before this drug the compound is taken over, compound combination. And then right 24 hours, you see clear evidence for synergy. But the important point is here, it's all within the same framework of Gaussian processes, right? Nothing ad hoc. Okay, I think that's it. I want to summarize what I've done so far. I think I'm nicely on time, right? Yeah, taking half a minute for this. We have developed a new null reference model named the mean model. We show that it performs better on 2 data sets with ground truth in brackets, not provided by us. We've piloted the first step to generate a benchmark data set for predicting synergy. That's ongoing work. And now we're in the process of developing a new approach to synergy based on Gaussian processes that provide a unified framework for dose and time dependence of synergy. Now, obvious current future plans, as for now, is of course finish collecting those benchmark data set. Something that Julia has largely done, but I didn't discuss about, is including monotonicity in GP models, right? You can do that. So far, we've not done it, but the figures I showed, I think, actually had it in it already. And then, of course, we have to benchmark our GP model just like we did with the mean model. And that's it. Thank you for your attention, and I'll take questions now. I have to stop my share, right, I think.
Thank you very much, Dr. Dijkstra, for the very informative presentation. So now, ladies and gentlemen, we have come to question and answer session. Before I start taking questions from the chat box, if you want to ask verbally the question, I now open the floor for any participants. You please feel free to unmute yourself and proceed with your question. Else, I think we can just go straight to the some of the questions that we receive from the group chat. For question number 1 from Dewi, Miss Dewi, she is asking one of the problem with GP is the computation time. So she wants to know your experience in applying GP in such data set.
Okay, yeah, that's an excellent question. Indeed, GPs computation time is an issue. I think what I showed here, this thing takes around 10 seconds on a very decent laptop to fit. And it's, I think, 8 by 8 doses and around 20 time points. Now, one advantage, you could say, of this type of setting is that the data is sizable, but also not that huge, right? I mean, going overboard with an experiment, 12 by 12 doses and maybe 50 time points, that would be an incredibly huge experiment in this setting. And even that is doable with a GP. Adding a third compound, I've already never seen, right? Because the costs simply just multiply, right? So that's also why GP is probably a good fit. That I don't see these data becoming really huge, right? You have in some other settings. So very good question, but I think manageable already now, and doable definitely in the future, right?
All right, hopefully that answers your question, Ms. Dewi. We also have another question from Fajar Agung. Actually, two questions from Fajar Agung. So the first one is how to define the best 2 or more drugs to choose for a particular disease, and is there any standard or guideline to follow? And if we can cover the second question, can we add patient conditions like blood pressure or glucose level into the formula in order to have a more personalized medicine? So that's a 2 question from Fajar.
Yeah, that's 2 questions. Okay, that's first, the second one. Yeah, I mean, this approach is developed for screening, right? That's our background. So essentially cell lines, right? That is the big background. It would be a whole other step that has been very little piloted to include, to actually do that in patients and develop personalized medicines based on this, right? So far, the drug combinations are just empirically established, right? There's databases where people have seen adverse effects, but to my knowledge, there's very little modeling. It would be, yeah, big conceptual. It would be a big effort to apply this to real patient data. Having said that, I think the Gaussian process model is then more flexible than any other, kind of fixed framework to do that. Right? Of course, you can include categorical predictors or even a smooth predictor like blood pressure or heart rate into some of these models. But boy, that would be an effort. Yeah, it was exciting research. What was the first question?
The first question was how to define the best 2 or more drugs to choose for a particular disease. And is there any standard or guideline that we can follow for that?
No, not that I know of. Yeah, I think this is really, again, this is what I've been doing is kind of, you could say, fundamental research or basic research, right? And the step from cell lines to real patients is a big one. So there are, of course, guidelines on drug interactions from the FDA, right? That's what was used here, kind of in this thing that I use as a motivation example, right? But, right, I mean, while these effects are indeed statistically significant, I mean, the predictive value of it is really weak. I mean, I don't know, top of my head, but maybe Simone is in the call, she can correct me, and the predictive value wasn't very high. So area under the curve of 0.8 or even 0.7. It's not like something you can write home about. So yeah, I'm afraid we're still far off there.
All right, thank you for answering the question, Dr. Dijkstra. I think it provides actually an opportunity for a new research area for the second question at least, right?
Yeah, definitely. Yeah.
Right. I think we have time for one more question. If we have, let me just recheck. Yep, we have one more from Yuan Hu Gu. He's asking, actually he's giving a statement here, since there are no specific drugs for the COVID-19, there are many treatment options on the market. And his question is, do you have any plans or ideas to apply your method to research the treatment of the new COVID-19? Very interesting question here.
Yeah, very interesting. But yeah, I know that definitely the screening for the so I think so. This where was I with this thing? So the L10000 for a while. That's part of the reason why we actually haven't done step 2. I think is that for a while the facility was exclusively being used for treating for kind of screening for COVID compounds. So we couldn't use it. We were set back. Obviously, that had priority, right? Ours is just a nice benchmark data set, whereas the other research obviously had priorities. I actually don't know what came out of it. I think the screening facility just got. I mean, that's what you often do in industry. They get the compound from companies, and all there is a number on the file to identify it. They actually don't know what's in it, and just run the screen. So they didn't even know what actually it was or if it helped. So no, I have currently no plans. I would have to have a partner with whom to do that. And I think just finding single drugs that work is already a huge issue. And then I think compound combinations is probably something for the far future with respect to COVID. So I'm sorry, no plans.
All right. Yep, he is thanking you for that response. All right, I think that's all the time we have. Let me just check for the last time. Yep, that's all the questions that we have. Thank you for the explanation and answering the questions, Dr. Dijkstra. We can see that the contribution that had been presented just now can possibly make a very great impact to the body of knowledge. And I believe that this provides a lot of research opportunities and hopefully continue to draw immersive attention from researchers, as well as, pharmaceutical enterprise to invest in ongoing as well as future research.
Yes, thank you. Well, thank you for chairing the session.
No worries, ladies and gentlemen.
At the beginning, it was a bit rough, but I had to get used to use Zoom. So it's my first time, so to do it this way.
No worries.
Thank you very much.
That's always first time for everything. So we would like to say thank you again for Dr. Tjeerd Dijkstra. I really hope that I pronounced your name right. So
you did a very good job.
Thank you. So we finally come to the end of the presentation. It was a very informative and interesting talk, and it was a pleasure having you with us. And I would also like to thank the audience for the active participation. So this concludes the keynote speech for this session. Thank you for attending. We hope that you have learned and enjoyed this session. Till we meet again next time, have a great weekend, everyone. Assalamualaikum warahmatullahi wabarakatuh. Now I return the session to the Madam MC. Thank you.
Thank you.